Hey guys, I'm just going to talk you through some of the things you need to know from Key Area 1 of Unit 1, the structure of DNA. It's basically going to look into the structure at greater detail than you learned in National 5, and then we're also going to look at how it's organised in different organisms. DNA, deoxyribonucleic acid. It should not be new to you. You've done this in National 5. You should already know that it is a double-stranded helix, that it is made up of nucleotides, which have a sugar, a phosphate, and a base. There are 4 types of bases: adenine, thymine, guanine, and cytosine. They also have a special base pair rule, where thymine always pairs up with adenine and guanine always pairs up with cytosine. This should not be new information to you. Okay? Right, nucleotides. From National 5, you already know they're made up of a phosphate, a sugar, and a base. We're adding the term in deoxyribose sugar. That's just the type of sugar it is. And this diagram here you should be familiar with. It's showing our nucleotide. Now, we're going to add some more to this. We're going to add some directionality to the nucleotide. Okay? And it's in the form of the term 5' end and 3' end. Let me explain what that means. Right, so what we've got here is a diagram of a nucleotide showing its chemical structure. Now, if you're not too sure about chemistry, what you need to know is that every point on the diagram, if it's not got a letter on it, it's indicating that it's a carbon atom. Now, the 3' and the 5' is actually all about counting how many carbons are on the sugar. So, to the right, we've got our first carbon, second carbon, third carbon, so our 3' end of the nucleotide, and it's this carbon that the phosphate of the nucleotide below joins onto. Okay? Then we get up to our fourth carbon and then our fifth carbon. And the fifth carbon is where the nucleotide's own phosphate is joined onto. So what would happen if we click on the next slide, it'll show you how the phosphate from this nucleotide is going to join on to the carbon 3, or the 3' of the next nucleotide. Right, so we're going to look at 2 nucleotides joined together, and you can see that there is a red line between them, and that is indicating our strong covalent bonds. Okay, a bond will form between the phosphate of one nucleotide and then the 3' carbon of the neighbouring nucleotide. And this allows us to join many nucleotides together. And we call it a polynucleotide chain, poly meaning many. And if we get the term sugar phosphate backbone, because these covalents are so strong, so it's meant to be strong like a backbone. Okay. Now, I'm just going to grab myself a wee pen. If we look to the diagram on the left, don't know my left or my right, on the left, you'll notice that one side it's got 5', and then it runs down to 3' here. Okay, that was meant to be an arrow. Whereas the other side, you've got 5' at the bottom, running up to 3'. Okay, they're running in opposite directions. We say it's anti-parallel. I'm going to come into that later because that's really, really important when you get into DNA replication. Okay. Okay, so let's quickly go over the chemical bases. You should already know this from National 5, but there are 4 types of base that a nucleotide could have. It could have adenine, thymine, cytosine, or guanine. And of course, when we've got our 2 strands of DNA, they join together in the middle with bonds between bases. Okay, adenine always pairs with thymine, and cytosine always pairs with guanine. If you struggle to remember this, there's a saying that some of people's like, it's at the golf course, so A joins with T, G joins with C. If you remember the words at the golf course, you'll remember the base pair rule. Now what we've got down in this diagram is a strand of DNA on its side, and you can see the A joining with the T here, and then the G joining with the C there. So we've got our base pairs. Okay. And the bonds that form between the base pairs are weak hydrogen bonds. And you'll heard this at National 5, the bases are said to be complementary to each other. Now, if you've ever wondered why does A only bond with T and why does G only bond with C, it is because of the number of hydrogen bonds. If you look between A and T, you can see there's 2 bonds. If you look between C and G, you can see there's 3 bonds. Okay, that's basically why they all pair together, because C can only ever have 3 bonds. So it has to have those bonds complete. So that's why a C can't join with a T, because then it's got a bond that's empty. So that's why A always bonds with T and G always bonds with C. So we're going to go back to the idea of having anti-parallel strands. Remember we talked about the 5' end or 3' end. 5' where they've got the phosphate, and then the 3' end where it's the bit of the sugar that joins on to the phosphate on the nucleotide below. Okay. If we look in this diagram, you see we've got our 5' on the left-hand strand up at the top, and then we've got our 3' on the left-hand strand at the bottom, and then it's the opposite on the other side. So you can see here that this phosphate is joining on to 3' here. But the reason it's anti-parallel is because on the other side the nucleotides are flipped over, they're upside down. Okay, so instead of having a nucleotide like this with a phosphate on top, we've now got nucleotides that have the phosphate on the bottom, but it's still on the 5' carbon, and we've still got the phosphate joining onto the 3' bit of the sugar. Okay, so it's still the same rules are applying about the 5' and 3', but the reason that they're anti-parallel is because on one side they're flipped upside down. Now if that wasn't simple enough, this bit round here summarizes what I was trying to say. It means that one strand of DNA has the 5' end at the top and the other has its 3' end at the top. So they're running in opposite directions, anti-parallel. That's where the name comes from. Right, so now we're going to go into the double helix shape of DNA, which is not new information. You know from National 5 that DNA is in the shape of a double helix. Okay, you just have to keep knowing that for higher. However, if you are wondering why it is a spiral staircase shape, this is not in the course or anything, but it's because of the chemical attractions between different parts of the nucleotides and different parts of the bases. That causes the DNA to spiral round. There's different parts of the ladders that are more attracted to each other than other bits, meaning that you don't actually have your equal spiral. And you'll see here that there's some parts are much longer curves than our other parts. Okay, and it's all about the chemical attractions. Right, usually during class I will have stopped every now and then, we'll have had some discussions, and then people will take some notes. Obviously we're not in class. So I'm just going to summarise the need to know stuff, and if you ever see a little pencil in any of the slides, that's normally the notes that a lot of my pupils would take. You can either watch the summary till the end, then go back and take your notes, or you can pause the slideshow as we go. Okay? Right, so you need to know that DNA is composed of a chain of nucleotides. Nucleotides are made of a phosphate, deoxyribose sugar and a base. And there's strong bonds that form between the deoxyribose sugar and the phosphates of the nucleotides. This forms the strand, which is also known as the sugar-phosphate backbone. You also need to know that there's 4 different bases on the nucleotide: adenine, thymine, cytosine, guanine. You need to know that the base sequence of DNA forms genetic code. I didn't talk about that, but you should be pretty confident that from National 5. And it's weak hydrogen bonds that form between our bases. And again, they might be complementary to each other. A pairing with T, C pairing with G. New information though, anti-parallel strands. The DNA strands have a 5' end and 3' end. 5' end is where the phosphate group is. 3' end is where the deoxyribose sugar is. 2 strands twist and coil, forming a helical shape, and it's therefore referred to as a double-stranded helix. Right, so looking at the success criteria for this key area, I have just talked you through the first 4, okay? The next tasks are going to involve some problem solving about our anti-parallel strands, and then you're going to look into, watch some videos about the discovery of DNA, and then the second section of the lesson is all about the organisation of DNA.